Good day, ladies and gentlemen. As introduced, my name is Taverna-Turisan. I'm from Omnia Fertilizer, where I manage our agronomic development and innovation departments. It's a great pleasure to be presenting to you today on my topic of how metabolomics is used to better understand plant biostimulant interactions, or a lack thereof, and then in turn, how this information is used to guide product development strategies, as well as how we incorporate these products on farm. Now, today's symposium has fittingly been titled 2020 Vision for Crop Production Under Climate Change. Over the past decade, numerous studies have been published indicating that significant and accelerating changes in local patterns of precipitation and temperature are to be expected for the 21st century across the globe. And due to its nature, agricultural productivity is vulnerable when it comes to climate change. We are well aware of the negative effects that abiotic stresses such as drought have on the productivity of our farmers. And these drought periods are only expected to become more prevalent. Now, the degree of this vulnerability will depend on both the biophysical impacts of climate change on crop yields, as well as our ability to adapt to changing production conditions. If we have a look at the two maps of South Africa attached, they represent 12 and 24-month standard precipitation index maps, which give us an indication of areas where prolonged droughts exist. They essentially highlight areas where below normal rainfall has occurred over a certain period of time. And if you look at the 12-month map ending May of this year, you can see in red the extreme areas where far below average rainfall was experienced. And for the 24-month period, we can see that we've actually been experiencing droughts for a prolonged period of time already. If we go further back into our timeline, this image will just become worse. To look at the dark red areas represent the extremely dry locations where far below average rainfall is experienced. Then we move on to severely dry, moderately dry, and somewhat dry. Now, the second and last study that I'll be showing you is a further study which highlights agriculture's vulnerability, where predictive modeling based on different carbon dioxide emission scenarios has been done. And they identified current and future climatically suitable areas for maize using this model for the year 2050 in comparison to our current scenario. So if we look at our current scenario, at the top we have our low yielding maize areas, and at the bottom the high yielding areas. These represent areas of 2.58 million for the high yielding areas and 2.95 million for low yielding areas. Taking a look at the conservative scenario in the middle, there is a 25% loss in the climatic suitability for both the low yielding and high yielding areas. While if we look at the pessimistic scenario, suddenly these losses are in the vicinity of 47% for low yielding areas and 30% for high yielding areas. And you can understand the implications that this has on food security, food production, with an ever increasing population. So these two simple yet telling examples of the potential effect that climate change may have on our agricultural production highlights the need to start modifying and adapting our strategies and approaches if we wish to keep up with the demands of agricultural production under climate change. And this brings us to the plant biostimulant interactions. Now, there has long been controversy with biostimulant products being referred to as snake oils and falling under the umbrella of pseudoscience. And understandably, this resulted in a poor market confidence when it came to biostimulants. To compound this problem, it was challenging to scientifically understand these mysterious products and their compounds and how they interacted with plants on a gene level. Furthermore, when applied on field, they often gave a mixed bag of results. Hence the importance of using innovative technologies to try and develop an understanding of the underlying mechanisms of these plant biostimulant interactions. Through the generation of knowledge of this kind and the application thereof, we are able to add value on farm and then continue to build market confidence in these extremely important products. As we move towards the latter part of the presentation, you'll see how we aim to integrate this in a more practical manner. Now, if we look at trying to understand these interactions, firstly, it's important to acknowledge that a plant's initial response to a biostimulant application occurs on a cellular level through differential gene activation, metabolic reprogramming, cellular signaling, cellular reprogramming, for example. This then translates into physiological traits, such as improvements in photosynthesis, changes in morphology, improvements in grain filling or carbohydrate storage, water use efficiency, etc. And finally, this translates into physical or agronomic traits, which we are familiar with and most often use in marketing data. This is the biomass, root biomass or yield or color, quality, etc. The challenge arises when we do not achieve consistent agronomic traits on field for unknown reasons. And it is these unknown reasons which cause the distrust in biostimulants within the marketplace. However, if we know and understand the underlying mechanisms of how these products work on a cellular level, it can offer valuable insights into further development and know-how of products, which can then be successfully incorporated into holistic management strategies. To do that, we can incorporate innovative technologies such as metabolomics into a comprehensive and systematic approach to better understand these underlying mechanisms, as well as to discover and characterize novel biostimulants. So what is metabolomics? Metabolomics essentially forms a part of the omics field of research. Just as genomics is the study of DNA and genetic information within a cell, metabolomics is the study of substrates and products of metabolism, which are influenced by both genetic and environmental factors. To state it simply, metabolomics is the large-scale study of small molecules, which are commonly referred to as metabolites, within cells, biofluids, tissues, or organisms. Examples of the metabolites that are analyzed for include glucose, different fatty acids, lipids, amino acids. Now, many reactions take place continuously within cells, so concentrations of metabolites are considered to be very dynamic and may change rapidly from one time point to the next. Current analytical techniques used to investigate metabolomics can only take a snapshot in time under a set of defined conditions. Now, if we have a look at a typical multi-step workflow of a metabolomic study, it would look something like this. You start first with your study design, planting, and application of your treatments, which then progresses through to the harvesting of different plant tissues at specific physiological stages, post-drought, salt, or moisture stress. Samples are frozen in time and then sent through to the laboratory where sample preparation and metabolite extraction is performed. Sample analysis is done using various techniques, including liquid chromatography or gas chromatography, mass spectrometry, as well as nuclear magnetic resonance, just to name a few. We then move on to data acquisition, where metabolomic studies generate heaps of data and can be quite intense to work through. And finally, we have our data analysis and data interpretation. Now, zooming into plant metabolites. Plant metabolites is a particularly interesting field because of the range of functions of both primary and secondary metabolites in plants. There were roughly 300 distinct metabolites that could be routinely identified per sample a decade ago, and this number has been gradually increasing over time. To give you a rough idea of the variation of metabolites between species, it is estimated that there are around 200,000 metabolites across the plant kingdom, and somewhere between 7,000 and 15,000 within an individual plant species. Now, abiotic stresses such as drought, salinity, or heat will bring about severe changes in the growth and development of plants, which in turn leads to the excessive amassing of secondary metabolites. Under prolonged or high-intensity stress, plant development is compromised. Plants experience a metabolic change, which then shows in the phenotype. Visual symptoms typically appear, and if left untreated, could be fatal or cause yield loss in plants. Through using metabolomics, we can measure which metabolites plants are producing in response to a specific stress at a specific time point. And ultimately, this information is used to guide product development processes in order to create products which are designed to help plants fight specific stresses. Now, if we look at the plant's adaptation to stress, plants undergo a genetic and metabolic adjustment in response to a stress. This is commonly known or referred to as the priming effect, which requires both time and energy. Much like how a flu leaves you without energy, your energy is redirected towards fighting the flu. The plant's energy is redirected towards fighting the stress. This is the reason why most biostimulants applications prior to stress show the best results. Through the application of specific biostimulants targeted for specific stresses, instead of this energy being wasted through going through the natural priming process, plants are already pre-primed to adapt faster and thus better tolerate that specific stress condition, which ultimately results in the reduction in potential yield losses. The typical adaptation response is from primary metabolites through, which is redirected to, for stress response into either secondary metabolites or enzyme activity in the following metabolism. So why metabolomics? Well, in general, metabolomics presents us with a tool to understand molecular mechanisms underlying plant life or biochemical events at specific physiological stages. It also provides us with a predictive understanding as a tool to lead innovative agricultural strategies. More specifically, new biochemical markers for environmental stresses can be used to understand or explain environmental influences on crop quality, as well as plant-environmental interactions at a molecular level. And lastly, value is added through developing new strategies to aid plants in tolerating environmental stresses, be it either abiotic or biotic stresses. This leads to a scientifically grounded and knowledge-based approach on farm, which is the direction we have to be moving towards when it comes to these biostimulant type products. Now on to the applications of metabolomics, some examples. I'd like to share with you at a high level 3 trials which were carried out to demonstrate the application of metabolomics in understanding these plant biostimulants interactions in response to specific abiotic stresses, namely salinity stress, nutrient stress, and drought stress. The first study looks at a tomato salt stress trial, where a mineral-based biostimulant was applied prior to the salt stress, which then was shown to induce reprogramming of the metabolism towards salt stress alleviation. You can clearly see that there are clear distinctions between the different treatments, being the control in green, the salt stress in red, and the soil-based mineral-based biostimulants, as well as the foliar-applied mineral-based biostimulant. This information can assist us in defining the optimum application methods based on evaluating the differences in the metabolic pathways and metabolites that are produced. We're still busy with this leg of research, which is using metabolomics as a tool to determine optimum application methods. Now, as we move on, this slide essentially shows the exact same data represented in a different configuration. As mentioned before, metabolomic studies generate huge quantities of data that are quite intense, and it takes a long time to work through them. Hence, with different chemometric modeling, we are able to further separate and refine this data into more simplistic visualizations. As you can see in this infographic, it is much more clear where we have distinct clusters between your stressed plants and your control plants, as well as between your stressed plants and your stress plants with a mineral-based biostimulant applied. Hence, we see here that this mineral-based biostimulant has activated a chain of biochemical pathways, which include flavone and flavonol biosynthesis, fatty acid metabolism, arginine and protein metabolism, to name a few. This points to differential metabolic profiles, showing that the application of a mineral-based biostimulant formulation led to changes in metabolism towards salt stress alleviation. Moving on to the next study, we have a look at a MAIZE trial that was conducted under nutrient stress conditions, where humic substance was then applied. As with the previous data, this infographic shows the distribution of different samples based on the underlying metabolic profiles. In the case of this graph, we can again see two clear clusters representing two groups, which again show that the addition of a humic substance under stress has triggered a chain of biochemical events which moves the plant's metabolism away from the stress closer to the non-stressed. And the pathways used in the study that we identified were again arginine and proline metabolism, phenylpropanoid biosynthesis, carbon fixation photosynthesis, just to name a few. And finally, we have a look at the third stress, being drought stress. In this trial, maize was used as a test crop, and the biostimulant of choice was a plant growth promoting rhizobacteria biostimulant, or PGPR. Now, this graph highlights the impact of a PGPR on drought stress. One can see that through adding a biostimulant with no stress, there's a lesser effect on the metabolic pathway. Now, there were certainly changes in metabolites being produced under this condition as well, but they were not as drastic as under drought stress. We can see here again that there's a clear distinction between the metabolism between the control groups without stress and the stress group. It's also very clear to see that the addition of a PGPR prior to drought stress has a priming effect in shifting the plant's metabolic profile closer to that of the plant under no stress. Now, metabolite production induced by the plants include arginine, proline, phenylalanine, just to mention a few. Depicted in this graph is essentially a snapshot of the root biomass of such a trial that's undergone drought stress. It's very clear to see that through adding a PGPR, we in fact saw an increase in root biomass despite there being no stress applied. To better understand this graph, you can see that treatments which were exposed to the same level of stress are colored in the same pattern. What's interesting to note here is that adding a PGPR under low stress and even severe drought stress, shown as the third and fourth columns, showed no statistical difference to the PGPR with no stress. And in addition, the low stress and severe stress with no PGPR was significantly worse than all other treatments. This is exactly how we want to decrease risk on field. But more so, we now understand how the plant went about compensating for that maintenance of root growth under drought stress by understanding the actual metabolic pathways that it followed. That priming effect with our biostimulants is essentially what we're after and what we need to apply to field practices. Now, some of you may have noticed that there were a few overlapping underlying metabolic mechanisms that the plants used under various stresses with various biostimulant applications. Seeing that similar metabolic pathways are induced by different biostimulants, as well as a few unique pathways, will enable us to develop specific formulations of new products for specific on-field conditions and/or specific stresses. This provides us with a scientific basis for product development within a comprehensive field approach. So what now? Well, we've established that the application of specific biostimulants under specific stress conditions results in improved plant growth parameters. We've also shown how plants' metabolism is shifted and altered. We've also shown the potential tool to further understand plant biostimulant interactions where different pathways were reprogrammed, which were related to enhanced abiotic and biotic stress tolerances. So what does all this mean? Well, at the end of the day, it has to result in something more tangible than changes on a cellular level. That is, improvements in either nutrient use efficiency or water use efficiency, improvements in quality parameters of our crops, or this needs to then translate into yield increases, or at the very least, a higher profitability on farm. And this brings us now to the practical integration. We now have an understanding of how our products work. So how do we take this to the field and ensure that we get consistent efficacy? How do we increase the reputation of biostimulants at a farm level? And that brings me to soil health. So soil health is the fundamental starting point of life and therefore agriculture. Soil health is essentially the center point of congruence when the physical, nutrients, and biological facets of soil start working together. Components such as nutrient interactions together with soil biology has historically been ignored to a certain degree, or for example, water relations with root health status. The biology has always been the least understood, and understandably so, with soil being one of the most complex ecosystems ever. However, I think we have progressed far enough to evaluate and measure these different indicators of soil health to bring them into more of a balance or an optimum. Now, optimum soil health is pivotal in having a knowledge-based and comprehensive solution with precise inputs being recommended. What would happen, for example, if we were to add a highly effective biostimulant to a system where the most limiting factor is out of scope of that specific biostimulant? You would most likely not see any results. And furthermore, it's the same as saying, would you take a calcium supplement for vitamin D deficiency? Of course not. Hence, more precise solutions or recommendations within a holistic approach is where we need to start progressing to on a field level. Now, from a soil health perspective, identifying our limiting factors on an integrated and holistic method is a way to move towards a precise and targeted solution on the field. Many farmers think that a biostimulant is a nice to have on the side, and often when it comes down to price negotiations, they are the first thing omitted from a program. We have to try and move towards implementing programs where biostimulants have a specific and integral purpose in order to create value for them on the farm. If we explore soil a little more in detail, we can have a look at a survey that we conducted to better understand which factors of soil health were critical for high-yielding maize. And in building models through factor analysis throughout the life cycle of the crops, we found that by taking soil and leaf samples at just 4 weeks after emergence, we were able to determine with a high level of accuracy what the yield would be based on the nitrogen, phosphorus, potassium, and magnesium percentages. And I'm not sure if you can see clearly in this image of the pivot, but you'll be able to see by the little black circles where the samples were actually taken throughout the course of the growing season. Now, nitrogen, phosphorus, potassium, and magnesium being able to determine the yield is not new. This isn't news. But what is fascinating, though, is that at the same time point, at 4 weeks after emergence, urease activity in the soil, together with total plant parasitic nematodes, were also dictating yield at this point. And therefore, I emphasize the importance of soil health in a holistic approach, that takes a number of different factors into consideration. Here we have another example of an integrated model. On this specific pivot, we have the differences in elevation in the drainage and terrain model, as can clearly be seen, which correspond to a nematode problem in samples that were then subsequently taken during the growth season. Now imagine adding a biostimulant or biocontrol product to a system like this, where the primary problem is not nematodes, but is rather drainage. Under such a situation, we would not expect to see the desired results, but this does not mean that the biostimulants now have to lose credibility. They are simply being incorporated into a program incorrectly. Again, this points towards the importance of a holistic and integrated approach to trying to incorporate biostimulants in a more practical manner on a farm level. Now, as I conclude, I'd like to reiterate the importance of soil health, which is the fundamental basis of life. We cannot look at product development in isolation. The application thereof is probably more important to achieve success on field. This can no longer be a hope strategy, but rather a strategy which is holistic, knowledge-based, comprehensive, scientifically grounded, and last but not least, measurable. I'd like to make acknowledgements to two of my colleagues that have contributed massively towards the above mentioned studies, Nessa Woodley and Dr. Fidel. Thank you for listening to the presentation. I hope that you found it informative and that I didn't rush over anything too quickly. It's a completely different experience presenting to a laptop and recording yourself versus speaking to people in person. I look forward to answering any questions that you have, and I hope that you enjoy the rest of this symposium. Thank you.